So my name is Brenna McKinney.
Alex Hardy.
I'm Molly Mayo.
I'm Sally Gamble.
Katie Barnes. And we did our presentation on the elimination of problem behavior by providing reinforcement for alternative behavior. So while designing this experiment, Dr. Reid had asked us what all of our future career interests were, and a common theme arose among all of us that we were interested in helping people. So we all did our literature reviews on the elimination of some type of problem behavior. So for example, I did mine on eating disorders.
I did mine on substance abuse, focused on alcoholism.
I did mine on post-traumatic stress disorder.
I did mine on heroin and opiates.
And I did mine on burnout.
So this experiment was designed to learn more about these problem behaviors and to better understand how to help people through the elimination of the problem behaviors.
So what we observed is that in these ways of helping people, the interventions follow a three-phase procedure. So first, there's the identification and the measurement of the problem behavior. Next, an intervention is introduced to eliminate the problem behavior. And finally, there's an assessment to discover whether or not the problem behavior was eliminated. So as you can see, this would be an example of a problem behavior, which is alcoholism. In the second phase, the intervention is introduced and the person no longer has any access to alcohol and other reinforcements are available. In this last phase, the person is out of rehab and they are exposed to the same cues as before the intervention. Now if the person starts drinking again, this is called relapse.
One problem with the three-phase procedure, though, is we still see relapse. One form of relapse that we focus on in our study is known as resurgence. Resurgence is when a problem behavior has been eliminated, however, comes back on the removal of reinforcement for an alternative behavior that has replaced it. Differential reinforcement of alternative behavior, or DRA, is commonly used in problem behavior elimination research. DRA is when an alternative behavior is gaining reinforcement while the problem behavior is on extinction. In this study, as well as many studies in this area, we utilized pigeons. Using animals for research focused on humans is considered translational research. This kind of research allows for more ethical experiments, and the pigeons are utilized because they exemplify many of the same behaviors as humans do in this field.
Okay, so right here we observe a clear example of resurgence, and this is taken from another study, but we just wanted to give you guys an example. So during phase 2, an intervention procedure is presented. Therefore, the problem behavior is placed on extinction. The responses decrease substantially. And however, during the phase 3, we took away the problem behavior, and the problem behavior returned. And so our question is whether we can prevent this problem behavior from returning after an individual returns home to their natural environment.
So this figure is a multiple schedule design with 3 different components. So each session began with a 1-minute blackout where the lights were on in the box, but the pigeons were not able to peck any of the keys because they were blacked out. After this 1-minute blackout, one of the 3 components was chosen at random. So after the component is chosen, it runs for 3 minutes, and then there's another 1-minute component. So each pigeon was exposed to all the 4 components, all the 3 components 4 times for a total of 12 components. And the pigeons learned to differentiate between the components based on the placement of the lights. So as you can see, this component shows the target behavior, which we are trying to eliminate. And one way to eliminate the target behavior was to introduce another colored light. So in this component, the pigeons were reinforced for pecking the red key, and in this component, the pigeons were reinforced for pecking the green key. So we had 2 different independent variables that we manipulated, and those manipulated the variable interval schedule. And a variable interval schedule is the rate of reinforcement. So for our independent variable between groups, we had a rich group and a lean group. So for the rich group, they were exposed to a variable interval 30-second schedule, and this means that on average, the pigeons were reinforced every 30 seconds for the target behavior. The lean group was exposed to a VI 120-second schedule, and then our second independent variable was the DRA component. So there was a DRA high component and a DRA low component. So for the DRA high component, they were exposed to a VI 30-second schedule, and for the DRA low component, they were exposed to a VI 120-second schedule. So based on this, we had 2 hypotheses. First, that the pigeons in the rich group would extinguish more rapidly and resurge more quickly than the pigeons in the lean group. In addition, our second hypothesis was that when the pigeons were exposed to the DRA high component, they would extinguish more rapidly and resurge more quickly than they were exposed to the DRA low component.
So it's important to note that when assessing the intervention for a human problem behavior, the problem behavior is already established. Therefore, to accurately assess our hypothesis, we had to establish a problem behavior within the pigeons. To do this, for phase 1, we created this problem behavior by training the pigeons to peck the illuminated light key that was presented to them in each one of their individual boxes. So as you can see, for the no treatment component, the left key was illuminated for the DRA high, the middle key was illuminated, and for the DRA low, the right key was illuminated. There was no difference in the illuminated light key placement between the rich and lean groups. So the pigeons were expected in this phase to peck the white illuminated light key that was reinforced by either a VI 30 or a VI 120 second scheduled reinforcement, and the number of pecks was measured across 30 sessions.
In phase 2, the intervention phase, the white key is illuminated in the same spots as it was in phase 1. However, in the DRA high component and the DRA low component is now under extinction, but it is not under extinction in the no treatment component. The newly introduced alternative option is the DRA high, and it's a red key light on a VI 30 second reinforcement schedule, and a green key light on a VI 120 second reinforcement schedule. These are introduced. This phase introduces the DRAs in a high component and in a low component, accompanied with the extinction of the white light in an attempt to eliminate responses to the white light or the problem behavior. As stated earlier, we predicted that responses to the white light are going to extinguish quicker in the DRA high component versus the DRA low component due to more frequent reinforcement given in this component.
So for this phase, we extinguished responding by eliminating reinforcement for the pecking of any of the lights that were presented to the pigeons. As you can see, the illuminated light placement was identical to the second phase. Number of pecks was measured in this phase. However, this phase only lasted for 10 sessions, as you can see. So what we were looking for in this phase was an increase in the pecking of the white key, which was, as you can remember, the problem behavior that was presented to them in phase 1. So if an increase in pecking the white key was present, then that would indicate resurgence.
So this depicts the difference in responding between the rich and the lean group. And so if you look here at the far left panel, this was phase 1 or our baseline phase, excuse me. So this is where the problem behavior was established as represented by these 3 lines. And these are our target keys, so the white keys that the pigeons were pecking. So moving into phase 2, this is our intervention phase. So this would be where we are trying to eliminate the problem behavior. So the original target keys were all placed on extinction except for the no treatment components. As you can see, and that remained relatively stable. But here, the DRA high and the DRA low components were introduced as represented by the red and green lines here. So this would be very similar to a rehab situation where the individual comes in and the problem behavior is going to be extinguished, but a new or alternative behavior is going to be reinforced. So that's what we're hoping for. So then moving on into phase 3, we wanted to assess if there was any resurgence or not. So we eliminated reinforcement on all the keys. And you can see they all extinguished pretty heavily. So where we would find resurgence would be in one of these 2 components, because that was the original problem behavior. And as you can see, we found no resurgence here. And we juxtaposed it with this figure right here to kind of show you what it would have looked like. So for here you can see that responding was at 0, but upon the 3rd phase it increased quite heavily, which is not what we found. Between phase 2 and phase 3, responding was pretty much non-existent.
So this figure shows the individual pigeons that were averaged in the previous figure. As you can see, all the individual pigeons have the same pattern as the previous figure. As Brenna said before, resurgence would be shown right here, and there's no resurgence in any of the pigeons. So one way to calculate resurgence is to calculate the proportion of baseline. Proportion of baseline is calculated by taking the target behavior in phase 2 and dividing it by the target behavior in phase 1. In addition, the target behavior in phase 3 is divided by the target behavior in phase 1.
So this figure here depicts the proportion of baseline. And as Sally said, what proportion of baseline allows us to do is to measure the amount of responding in phase 2 and phase 3 to the original baseline amount of responding. So it just lets us equally assess it. As you typically you'll see a substantial amount of responding in the first phase, and then you just need a way to be able to compare them equally. So here you can see that responding did decrease, and that there was no resurgence, as opposed to here, where if you look at phase 2 and phase 3, there is a substantial amount of increase in responding. So if you look at the square right here, that is roughly 40% of what it would have been in the first phase. So this is very clearly resurgence, whereas we found actually a slight decrease in responding, so we can say with confidence that we did not find resurgence.
Okay, so this figure indicates that we observed no resurgence for each individual bird in both the rich and the lean group. And we calculated this by the proportion of baseline, and this was calculated by subtracting responses within phase 2 from phase 3. And we observed negative values, which indicate that problem behavior was even lower than before, which is the goal of designing a successful intervention. Okay, so this figure was taken from an experiment which we designed our experiment off of. And again, they have positive values, and that's indicating that phase 2, their intervention was not successful, and they did have resurgence.
So while we were very excited that our intervention was successful, we're not sure why this was the case. Based on all the research that we had done prior to and during the experiments, we very much expected to see a significant amount of resurgence within the third phase. What we do know is that, like Molly said, we based our study off of the Nevin 2016 study, but ours was a simplification of this study. So whereas we only had 3 lights, they had 5 different colored lights. And for us, each colored light corresponded to a particular component, as opposed to Nevin, where they kind of randomly assigned the lights. So we do understand that there was something about the simplification of our procedure that led to no resurgence, but the next step is going to be designing an experiment to really kind of tease this out so that we can better understand, because this is very exciting for the research and resurgence and helping people with these kind of problem behaviors. So we would like to thank Dr. Reid for his help and mentorship on this project.
All right, questions? Any questions? All right, thank you. Thank you to all the speakers.